Welcome to Bioinformatics.Expert. In this video, I want to show you how you can align the reads of your FASTQ file to a reference genome. In the last video, we used Trimmomatic to chop off some bases from the 5' side of each read in our file. And today, we want to use that file in the alignment. We will do the alignment with the program Hisat2. You can find it in the analysis section. Click in the RNA sequencing table on the Hisat2 button to start it. Here, select the output file from the Trimmomatic approach and continue. Hisat2 is also able to align FASTQ files from a paired end experiment. However, since our reads are only single end, we skip that for now. On the main page of the program, we see that we can select from the parameter table the reference for the alignment. We also see that we can choose if the DTA flag should be set in the prompt or not. The DTA flag is of importance if we want to run the program string t afterwards with the output file. Let's select Homo sapiens version GRCH38 as a reference because the experiment was made with human tissue cells, and select that we don't want to add the DTA flag. When we start the analysis, we have to wait a little bit now because alignment is the most time-consuming step in an RNA sequencing experiment, especially with Hisat2. There are faster programs, like for example the star aligner. However, this one needs a lot of resources. Nevertheless, I plan to eventually implement it later. After we have accelerated the analysis a little bit, we see that the results are actually quite good. We see an overall alignment rate of 96.36%, and we see that 86.12% aligned exactly one time. I would now suggest to test what the output of the alignment is when we directly use Hisat2 on the raw data FASTQ file. Because actually Trimmomatic is an optional approach. Of course, when you use Trimmomatic, you can increase the quality of your reads, and this might lead to a better result of your alignment. On the other hand, when you use Trimmomatic, you reduce the length of each read. That means that the alignment algorithm might detect more reads which not only bind to one region of the genome, but to multiple regions. So for those reads, we cannot be sure which one is the correct aligned region. At the end, you have to decide by yourself if you want to use the Trimmomatic or not. I want to show what happens when you don't use a Trimmomatic beforehand. So I start now Hisat with the same parameters as we had before. However, this time I use as input the raw data FASTQ file. Okay, so let's fast forward again a little bit. Now we see that we actually have an overall alignment rate which is quite similar to the one of the trimmed FASTQ file. With 96.43%, it's only 0.1% higher than in the run before. However, when we look at the exactly one-time aligned reads, we now see that we have a percentage of 87.39%. That means the alignment rate is about 1% higher than in the run before. Actually, this is as expected because, as I said, due to the lower read length in the trimmed FASTQ file, we get higher multi-mapped reads. That means for this data file, I would prefer to not trim the reads beforehand. However, other experiments might benefit from trimming. This needs to be evaluated for each experiment individually. In the last part of the video, we can now check what happens when we use Hisat in a different program version. I now start the alignment with all available Hisat versions. And we accelerate the analysis a little bit. After each run finished, we can now take a look at the results. So as said earlier, Hisat2 in version 2.2.1 has an overall alignment rate of 96.43% and an alignment rate of reads which exactly align one times of 87.39%. This is actually quite good. When we now go to Hisat2 version 2.2.0, we actually see exactly the same result. Hisat2 in version 2.1.0 has a slightly reduced overall alignment rate of 96.37, but an almost identical alignment rate of reads which exactly align one times of 87.37. However, when we now check the results of Hisat2 having an older version, 2.0.5, we see also the overall alignment rate is still very high with 96.30%. The alignment of reads which exactly align one time is only 80.37%. Here we have a drop of 7% of the reads which might not be used for downstream analysis. Also checking Hisat2 in version 2.0.4, we quite see the same results as in version 2.0.5. This means if you want to perform reproducible data analysis, you should stay at your Hisat version, because if you analyze data sets later with a probably newer version of the program, you might get quite different results out of them. And these might then lead also to other downstream analysis results. Okay, thank you for watching this video about Hisat2 and how you can use it in bioinformatic tools online. I hope you agree that you have to carefully select your program version to fit, for example, earlier analysis if you want to do reproducible data analysis. If you like to use that tool in your research, write me an email or visit my webpage.